Uh, Hello everyone. Good morning to all of you. And first of all, I'd like to welcome all the scientists from various levels of CSIR. So today, as we many of us aware of the CSIR program as a national level program, uh, key objective uh, to extend our classroom learning as well as uh, to increase the scientific approach among the teachers and students. So today under this Let's Talk Science Series 2.0, uh, CSIR-CSIO is organizing a webinar with a collaboration of NIYS, uh, Chandigarh chapter, and so for that we have a guest lecturer, Mr. Suman Tewary, scientist of at CSIR Chandigarh, who will be delivering a lecture on microscopy and applications. Um, so before I hand over to Sir, I would like to inform you that our program is live at CSIR Jigyasa YouTube channel. So anyone having any doubts regarding today's webinar, you can ask by commenting on YouTube and also in Meet also. So thank you, and Suman sir, over to you.
Thank you. Please share my screen. Am I audible?
Yes, sir.
Yes. Today we'll be discussing about microscopy and applications. So I'm working as a scientist in biomedical applications, CSIR Chandigarh. We're working in the different domain of medical images. I'm focusing my lecture on the application of microscope in the biological sector. This microscope, students in the school, they have seen the microscope in their lab. So basically, for any object which is very small, which we cannot see in our naked eye, that can be seen through a microscope. That is being used across different domains of science, and basically this microscope has given as the most useful device in across all the sector of science. Okay. So it was first developed in around 1590, and later on with different companies coming up, they have given different designs of microscope. So now it is in the domain of digital images also. So that is how the pathology which is being done for this microscopic imaging of different samples like blood, tissue, and other samples. So that is now becoming digital with the advantage of this microscopy as well as the camera and other systems. The anatomy of a microscope. So what happens like of light to go through the camera tube, where we put a camera to get a digital image of a sample under study. So using this setup, we can get a digital image of our requirement, which we can share among different pathologists, or even you can email, or we can have a digital platform for this kind of microscope. On the right side, if you see, so there is a sample slide, which is of a tissue sample. If you see, there is a small square made, and on the right side is the magnified view of that sample. So it is observed under 20x magnification. So I have shown a pictorial representation of this square. Basically, this area is even smaller than this small square. Pathology, basically, it is a domain of diagnosis. So where different diseases are diagnosed using blood, tissue, urine, or other samples, and it is basically a gold standard for disease diagnosis. So what happens here? So sample is prepared, and the sample is stained, and the slide is preparation, and that slide is kept under the microscope, and that is observed through this eyepiece or through the digital images as we use from the camera, and that image is used for diagnosis and inferencing. There is another terminology called telepathology. Basically, what is happening here? we have this sample on microscope. So that is kept at some other place, but the pathologist is sitting on some other city. So he or she can actually observe this sample under this microscope sitting in his or her office from the remote location. So basically, it is giving remote access to the microscope along with different control. So with this, we can have we require that high speed communication uh, for that purpose. So when, if the sample is kept at the remote village, that can be accessed throughout the country or anywhere in the world. So basically, what is happening here? So we have a basic pathology at a local paramedic staff. That he or she can put the sample in the microscope, and the microscope is connected through high-speed internet. And that remote access of this microscope can provide digital data, which is observed by the pathologist sitting in another city, and that pathologist can give the diagnosis or inferencing. So basically, yeah, uh, in Indian context, why we need this kind of setup, digital or telepathology? Because there is huge pool of patients, and there are a lot of blood disorders such as malaria, anemia, and other diseases which are very common, and that diseases require very fast and accurate diagnosis. On the other hand, cancer, which is increasing in a very high rate, and this is very much alarming situation because in India we have around 14.3% of all cancer incidents in the world. And also to diagnose this kind of disease, there is limited number of doctors. So we need a system which can assess these doctors to provide this diagnosis rapid and in a convenient way. So what I have shown earlier, so that was a single field of view of the microscope under one objective. But that many times it requires that whatever microscopic images we need to see for the assessment of the any diseases that may or may not be available in the single field of view. So what happens? Uh, uh, the pathologist need to move the slide in XY direction to see different field of views and then find out if there is any abnormality. But he or she may lose some of the information because it is very much impossible to get across the slide at each and every locations. So in those cases, we require slide scanner, which is basically robotic or automated microscope. So that can acquire the images while moving the XY stage. So the stage moves and that images are stitched to create a large field of view where all the images are stitched to get a complete view of the slide. It's very complete magnified view of the slide. So that company was Bliss System, which introduced the system in 1994, and now it is available as Olympus Bliss. So why these things are required? Because I already told increasing this number of cases of cancer. So this is the statistics of increasing number of cancers in India. So if you see, there are roughly more than 10 lakh cases of cancer reported every year. So it is day by day it is increasing. Also, because of this cancer, there are many other factors which is involved, like uh, financially or issues, then medical issues, as well as the economical condition of the country. Because if so many patients are there in the country, so obviously we need some setup to handle those large pool of patients. So that is why we required some kind of assistance to the pathologist for this kind of screening of diseases. Another was like blood disorder. So as we know that anemia is very common in India, and about 39.86% reported uh, cases of anemia in the world, whereas in other countries like Canada, it is only 3%. This is another report from Hindu. So that shows that alarming situation of this kind of diseases. Even some other diseases like malaria, sickle cell anemia, all these things are very common. Even sickle cell is common for the Adivasi populations, which is reported here on the right side, Kerala, Andhra Pradesh, Telangana, Assam, and all those regions. So we need to take care of this screening of these diseases using a microscope and with a rapid and accurate way so that this reporting can be done and preventive measures can be taken up. So there are some point of care diagnosis also available. So like basically blood tissue and other biological sample are collected, and there's a stain of the sample under the study, and that is kept under microscope. And we take different region of interest for the quantification of diseases. Many a times it is manual because manually pathologist observes the samples, and that is many times time intensive and always tedious because you need to uh, see different regions of that sample in a very limited time. So what is the need? So the need is to automate or diagnose digitalization of the sample under study, and also automated quantification of disease parameters for fast and accurate diagnosis. So how it can be done? So basically, if we use the camera, we can get a digital image. So there is this way of the image analytics pipeline, which can be used for this kind of screening. So first of the parameter is this image color space selection. So basically, that image which we see in a camera, or if you see any image, any color image, that is basically RGB, red, green, and blue. The combination of red channel, green channel, and blue channel. So we need to we can extract this color channel into different color space also to get different representations like hue, saturation value, CMYK, cyan, magenta, yellow, and black, like that. So this color space can give different perspective of the sample under observations. Then we can apply segmentation. Basically, segmentation is like finding out different regions within an image which is of our interest. So basically, if you see that on a microscope, under the microscope, we find different kind of cells, and we can detect those cells by removing the background of the stain. So that is done using this segmentation. So it will take out only the cellular regions and discard other background regions. Then we can apply some morphological operations to get proper shape of the cell. And then we can use the mask. So basically that mask will give you, take out the only those color information from the original image to get your output, desired output. And then we can apply different machine learning and deep learning applications for automated reporting. So before we going to this discussion, let's understand what is an image. So basically, image is a 2D matrix which is containing the intensity values. So if you represent an image in the form of fxy, so a different location xy represent the different pixel locations. Okay, so at different pixel location that f00, f01 to f0 m minus 1. So these are different intensity values at different locations. So what happens when we use a color image? So we have three channels, separate red, green, and blue. So all three channels will have separate intensity values. By combining these intensity values, we can get a color image. So we can apply different algorithms in the appropriate image channel to extract the defined features. So as I told you, like the color image combination of red, green, and blue that is shown here. So if you see in the left side of the image, so that image of a parrot, and if you see RGB, so different red channel, green channel, and blue channel representation, that is shown on the right. So since the parrot is having more red intensity, so the red channel, if you show, that is more bright compared to green and blue. On the other side, if you see, so that is a fluorescent microscopy where basically DNA dye, GFP cell line and tubulin are used. So blue channel, green channel, and red channel are used, and they are falsely colored. In the left side, it is like true color. We are extracting the red, green, and blue. On the right side, we are putting false colors to generate a color image. So that is those three channels are combined to get a merged image. Fluorescent microscopy. So as I said that RGB, those are basically additive color space. So combining these diff colors, we can get all the colors which we can perceive. And then CMYK is the subtractive color space. Basically, we can subtract RGB from one to get these channels. Then we have this hue, saturation, intensity. Many times we use LAV, XYZ, and other color space. So if we recall that in our old, like we had earlier black and white TV, so that is basically the gray channel. So that is, if you see in the image, so there is a color cube. So in the color cube, the center is black and the outermost point is white. So and you have RGB axis, and the combination of that gives you cyan, magenta, and yellow. And the diagonal from black to white is the grayscale. So those are the intensity values from 0 to 255 that gives us a gray image, which we earlier used in black and white TV. Similarly, for the color changes, we have also used hue, saturation, intensity for the colors, which is shown in the color cone. Now coming to that example which we have already discussed of the microscope. So if you see in this image, so basically the color image is having cells which is represented in a blue color, and some background other cells which is represented as red color. So the combination of red, green, and blue provides this color image, and on the right side we can see the grayscale image of the same color image. So when we use this clear scale, so we have only single channel. So that is basically combination of red and green and blue in different proportions to get this image. So all these images are showing intensity values in the range of 0 to 255. Why 255? Because we normally use 8-bit image, and for 8-bit image, the intensity value can be of up to the power of 2 to the 8 minus 1. So that means 258 is 256 minus 1, that means 255. So all the values will be intensity values from 0 to 255. Other representation of the same image in the form of red, green, and blue is shown here. If you see in the left half, so that same color image is shown in the red, green, and blue channel respectively. On the other half, if you see, that is for the same image, we are showing the hue, saturation, and value. So value is similar to the intensity of the image. Okay. So these channels are used for extraction of different information from the image. Then we'll talk about the image segmentation. Basically, from the image input image, we can take out different color channels, or we can use the grayscale image to segment out the region of interest which is required for our analysis. So here we have showing the two examples. In one example, we are showing the segmentation of the gray channel. On the other half, on the other example, we are showing the segmentation of red channel image. As we discussed earlier, so basically from that image, the red channel is extracted, and then we are segmenting that red channel to get the cells. So basically what is happening here, so segmentation is telling us, like, if from the intensity value we calculate that at any intensity value, if it is lower than that intensity value, we'll label it as 0. If it is above than that, we'll label it as 1. So basically that is a binary image in the range of 0, 1, or we can also say in the range of 0 or 255. So that if you see, all the cellulars were dark, so that's why those are shown in black color, and background is shown in white color. Then there is another approach where basically to extract different staining of a cell, we use color deconvolution. So color deconvolution usually extract the different stains, which is basically hematoxylin, eosin, and diaminobenzidine, or DAB. So in this specific image, we applied this approach to extract this DAB color space and hematoxylin. So basically, why these two are only there? Because these were blue and red cells. So normally blue is for hematoxylin and red is for uh, DAB. Red or brown, both are used for DAB. So if you see here, this hematoxylin is showing in all the blue cells in high intensity, and that all the red are showing here in the DAB stain of high intensity. And when we apply segmentation on this hematoxylin stain, we get only those regions which are having these blue cells. And similarly for the DA stain, only regions where we are getting a red cells. And when we apply masking, basically masking means we're using this set image. On the bottom, we have only applied to that DAB color space, and from that we are enhancing that. So all the cells are being detected here. On the right half, we are applying smoothing and sharpening of the image. So same image, after smoothing it, the edges are getting the effect of edges are getting deteriorated, and a smooth image is represented here. While on sharpening, the edges are sharpened to get a better representation of the image. So these are used for any kind of pre-processing before all this kind of segmentation and other approaches. Then we'll coming to this computational pathology. So basically, computation is the pathology which is having some kind of um, automated computation. So basically, this prefix of computation does not change the meaning of pathology itself. This aspect relates to the emerging tool or such as image processing and machine learning or deep learning to achieve the goals of pathology in new and perhaps better ways. Basically, what happens here, like all segmentation, classification, what is required, so that is done using this kind of computational tools which will assist the pathologist in decision making. If you see in the left half, so the cell nucleus detection in renal clear cell carcinoma, which is a case of cancer. So tissue microarray image was used, and all the benign cells are detected as and red are so the cancerous cells. So basically, what is happening here? So using some kind of machine learning and deep learning, we are actually detecting all the cells and then labeling it with different pseudo colors to assist the pathologist in decision making. On the other half, if you see, the computational pathology algorithm recognizes sub-visual morphological features and displaying alterations in the stroma that are associated with the ductal carcinoma in situ lesion. So here also it is showing a stage of cancer where this automated approaches is labeling different regions and assisting the pathologist to give decision. So all this computational pathology basically is a tool that can help a pathologist in providing influencing or score for given sample with an ease of use, because it will help them to reduce the time consumption. Also, it will be not tedious as compared to the manual score. On the other hand, there are some errors which is reported because of the subjective variation, because two different pathologists may or may not score the sample in the same way. But if you use the machine, that will always give the same score for the sample with much higher repeatability. So that is why this computational pathology is now being used across the world, and people are using different kind of image processing and machine learning tools to mm, score this kind of samples and assist the pathologist to give the final score. Machine can never give the final score, so we cannot be that is being used in the reporting. Otherwise, it cannot be used. So some examples which I'm showing here, these are the applications of microscopy. So here we are extracting the blood cells. If you see in the top, so there is a color image of a blood smear slide, which is having some cases of malaria. So in malaria, what happens? This RBC are getting affected, and the morphology of RBC changes, and always that texture as well as the color of the RBC changes. So if you see more or less all the cells are normal. There are few cells which are having of different shapes as well as color and textures. So these are basically infected cells which we need to detect. So what we're doing here, we're only detecting here cells. So we're applying some kind segmentation of the gray image, and after segmentation, we're having some kind of debris in the background. So those are removed using morphological operations of erosion and medial filtering. And after this step, you'll find that some of the blobs are having holes in that. So basically, within this white object, there are some black dots. So we need to remove that to generate a proper mask. So we are filling the holes here. So after filling the holes, we are getting the masked region. So we can tell that at what pixel the cells are located, and that is used for cell leveling. So on the last image, if you see, so all the cells are labeled with a bounding box. So whatever blobs we are getting in the previous step, so for the pixel location, minimum and maximum pixel locations are used to draw a square or rectangle. So from the pixel location, we can get that, and then we can extract these individual cells, and we can apply machine learning to classify each and every cell. So I'll discuss about this classification in the next slides. So here we're discussing the similar step like in the previous way, where all the RBCs are detected. In this step, we are discussing about RBC and WBC quantification with deep learning. So what is happening here? So we have trained the system instead of using the segmentation on all, we have manually annotated the images basically by drawing the cell area and then train our system. So from some test images, we have from some training images we have made different patches and so label it like RBC or WBC or different subtypes of WBC such as neutrophil, eosinophils, monocyte, lymphocyte like that. And then in the whole classification, those model is used to classify on new set of images. So we have used this network called YOLO. YOLO is known as you only look once. It will automatically make the bounding box across the cells and then classify it into RBC and WBC based on the training we did on the training images. So we have seen that on the test images, it is showing more than 90% accuracy. And this work was represented in International Science Festival 2019 in Kolkata. There's another example of ERPR marker. So basically, it is used for the cancer screening, where basically hormone receptors are used. So if you see here the input image, the input image is showing some cells which are in brown and blue color, and the image are obtained from two different microscopes. One is showing in bluish tint, another is greenish tint. So we need to apply some kind of approaches which can extract these cells and then quantify the cells of what class it is, as well as so there are two steps here, like one is like extraction of blue and brown cells, and after the extraction of the brown cells we need to quantify the intensity of brown. That means what is the intensity of brown color in those cells. So it may be so weak, moderate or strong. Means very dark, very light brown, moderate brown and dark brown. So dark brown means more positive and blue cells are for negative. So we have applied gray world normalization to normalize these images into similar color space. So if you see, so this bluish and greenish tint are normally normalized into this color gray world space. And here, although the second image is of low brightness, but still the color wise it is similar. Then we applied color normalization, segmentation, and cell area extraction, which you discussed earlier, like the blood cell images. And then after that area is extracted, we have applied machine learnings using artificial neural network, k-nearest neighbor, naive Bayes, decision tree, and random forest to classify the intensity of brown color, whether brown is light brown, moderate brown, or dark brown. So after that, we get this proposed output image where all the cells are labeled with pseudo colors. Blue shows the negative one, and yellow, orange, and red are for positive at different intensities. And that is also compared with one other software called immunoratio. And we can see that the detection of cells are quite better compared to the standard software. And also the scoring is matching with the expert score, which is having high correlation of more than 91% for the 50 cases. Similar. So far, we have discussed about only cell regions. So there is something called HER2 marker, where basically the staining is done on the membrane, cell membrane. So the boundary of the cell are getting some colors. So basically, if it is positive, my cancer is positive cell. So in that case, the boundary will be of brown color with different intensity based on the scoring. So if that complete membrane is there, when I mean by that, like the cell boundary is completely stained with brown color, then it will be higher positive. And if it is broken, some part, some membrane cut is broken, or there is no staining at all in the cell, then it is negative. So there are three classes normally we use, like positive and negative. And if that positive negative cannot be detected, we call it equivocal. So here we have applied the color normalization, segmentation, and morphological operations to extract the cell membrane or cell boundary. And from the cell boundary, we have applied pseudo coloring on the boundary as well as the detection of the cell. So if you see here, the image was there, so that is enhanced or brightened, and we have detected the cells, negative cells of blue colors, and the membrane staining of brown color, and then we have used skeletonization of this membrane to get the only the pixel location of the boundaries, and then we have applied machine learning, basically SVM classifier with Gaussian kernel to train from these patches to get what kind of score for the whole the image. And we have also pseudo colored wherever we get complete encirclement in this membrane, we label it as like red. If there is broken boundary, we label it with green color. And that we also compared this approach with immunomembrane software, and for this 90 images accuracy is very close to the expert score, which is 93%. So far, we have discussed about all the applications of microscopy in the biomedical applications. So these are some important points which needs to consider all the time. So there is no universal algorithm for segmentation of all classes of images, and we need to play with color spaces for suitable channel selection. Pre-processing and post-processing is essential to extract the region of interest, whatever we want to see. And then deep learning is an emerging area of study uh, which is being used in the digital pathology. Is there any question?
So, thank you, sir. And I'd like to ask the audience that if you have any question, then you can ask directly in Meet, or you can ask in YouTube by commenting them.
I couldn't find any comment in this chat box.
Yes, I am checking in YouTube also. Uh, Sir, there is no question in the YouTube also. Okay, sir. There is. I hope there is no questions. And thank you, sir, for your wonderful lectures. And I'd like to thank all the scientists from various labs and of CSIR, and all the scientists from CSIO also, and all the teachers and students present for this today's webinar. And thank you for making